Welcome to our video lecture on topic 2.4, proteins. Our essential understanding for today. Proteins have a very wide range of functions in living organisms. Here are some examples of those wide-ranging proteins. We have rubisco in plants, helps in photosynthesis. Insulin is a mammalian hormone that regulates blood sugar levels. Rhodopsin in our eyeballs helps us perceive visual information. Collagen is a structural protein found in our skin, and of course spider silk is what spiders use to build their most amazing webs. Our objectives for today are going to understand that proteins are chains of amino acids held together by peptide bonds. We're going to talk about how amino acids with different properties convey those properties onto their proteins, which is why we have those proteins that have various functions. Proteins have 4 levels of structure: primary, secondary, tertiary, and quaternary. Proteins can be globular or fibrous. This is all about their shapes. Proteins can be denatured. This is when we basically unshape the shapes. And then the function of a protein is related to its structure. Amino acids are linked together by condensation reactions. Remember that this is our set of anabolic reactions, the anabolic side of metabolism. We're going to link those amino acids together to make some peptides. Peptide is kind of a word that we use to mean like a baby protein. We could have a dipeptide that's only 2 amino acids. We could have a polypeptide with many amino acids. We could need to process those guys to turn them into functional proteins. Peptide bonds are what hold those amino acids together. And our dipeptides, our polypeptides, are proteins. Here is an example of a condensation reaction making a dipeptide. So here we have one amino acid, here we have another amino acid, and here we have a dipeptide. So here's the piece of amino acid number 1, the piece of amino acid number 2. We pulled out the hydroxyl group from one amino acid, the hydrogen from the other amino acid. These guys linked to make water, which is why we call it condensation. And then where this carbon was missing a bond and where the nitrogen was missing a bond, they're going to find each other to make a new bond. That bond right there is our peptide bond. Peptide bonds are bonds that hold amino acids together. What helps to form these bonds are ribosomes. Ribosomes, the protein factories of the cell. Ribosomes' job is to catalyze the formation of these peptide bonds during a process called translation. We'll talk about that more later. One of the things that IB loves to ask us is to draw an annotated diagram depicting the formation of a peptide bond between 2 amino acids. If you need to back up the video and look at that picture before we start to draw together, that's fine. But I'm not going to make you draw. We're drawing together. So we're going to start with 2 amino acids. Amino acids have an amino group. They have a central carbon, and they have a carboxyl group. So this is our amino group, N and 2 H's. We have our carboxyl group, COOOH. This is the carboxyl side of the amino acid. This makes it a carboxylic acid, which is why it's an acid. That's the amino part of the amino acid. Our central carbon is going to have a hydrogen, and then what we call the radical group or the side group. This is what's different from one amino acid to the other. This is what makes leucine different from cysteine, different from methionine. The rest of it, the amino acid, the amino group, the carboxyl group, the central carbon, those are all the same. So sometimes we say draw a generalized amino acid. This is a generalized amino acid. All of that is the same in every single amino acid. This is the only part, the R part is the only part that's different from one amino acid to the other. Let's draw another amino acid because we need to be able to bind these 2 together. So we're going to put those 2 H's on an N. Again, this is our amino group. Central carbon, hydrogen, R group. These 2 R's might be different, so sometimes we do R1 and R2 just to show that there are some options for different R groups, side groups here. And then we're going to throw our carboxyl group here on the end. So again, we have amino group here. We have our carboxyl group here. Central carbon in the middle, our group on that central carbon. All right, let's try to make ourselves a lovely peptide bond. So I'm going to pull the hydroxyl group off of the carboxylic acid group of one amino acid, the hydrogen off of the amino group of the other amino acid. These are going to combine to form water, which is why we call it a condensation reaction. And then what's going to happen is we're going to build a bond where we broke off those two bonds, and that's going to be our peptide bond. So what we're going to have is our amino group, central carbon. We're going to keep this carbon and that double O, carbon double O, but the OH is now gone. Instead, we're going to link that carbon of the carboxyl group to the nitrogen of the amino group of the next guy. It's only going to have one hydrogen left. This guy got lost and given away to water. So we're going to throw only one hydrogen, not two hydrogens, on that nitrogen. And then the rest of it is pretty much normal amino acid, central carbon with a hydrogen, and then our carboxyl group. Don't forget hydrogen on this guy, and then our R groups, and we could call them R1 and R2. And this is our dipeptide. To annotate it, we definitely should label our peptide bond. Amino acid, carboxyl group, amino group. So there are our annotations. We made our dipeptide with a peptide bond. All of our proteins are formed by amino acids being linked together with those peptide bonds and condensation reactions, but the number of amino acids that we put into proteins definitely is going to vary. Insulin is pretty small. There are only two chains of amino acids. One is 21 amino acids long, the other one is 30 amino acids long. So for a total of 51 amino acids, we have insulin, that hormone that helps us regulate blood sugar levels. Titin is in our muscle. It helps to hold this thick filament. It's called myosin. Helps to hold the myosin into our muscle fibers. Titin is a protein that is composed of, oh my goodness, 34,000 amino acids. So again, the size of our proteins can vary quite a bit. We mentioned a couple slides ago that the R group is what makes one amino acid different from the other. There are about 20 different amino acids that we use to build our proteins. There are more than 20 amino acids, but there's like 20-ish that us living things use to build proteins. Those R groups are what are different from one amino acid to the other and what give the specific characteristics to our different functioning proteins. So this amazing table is showing us some of those different R groups. Remember that our generalized amino acid, what they all have in common is an amino group, a central carbon with a hydrogen, and then the carboxyl group. The R group on that central carbon is different from one amino acid to the other. This table has the amino acids oriented a little bit differently. So the carboxyl group is what we have here, kind of at the top. The amino group is kind of sticking off to the side. So here's amino group, amino group, amino group. We don't draw all of the C's for all the carbons, so anywhere we have these points that don't have an element labeled, those are all carbons. So there's the carbon of the carboxyl group. This is the central carbon. And then the rest of this here is the R group. And then here's another R group. And here, look at the size of this one, also an R group. We also have some R groups that are super small. Glycine, here's that central carbon. Its R group is literally just a hydrogen. Kind of crazy sauce. So again, we have big variations in size. Some of our amino acids are charged, some are polar, some are non-polar, hydrophobic. Lots of variations in the chemical characteristics of these side chains allows our amino acids to be different and to build our proteins that have so many variations in structure and function. Having these amino acids with different polarities is especially important for our membrane proteins. Membrane proteins like these guys that literally embedded in our plasma cell membranes. And remember that our plasma membranes are composed of phospholipids. Phospholipids are hydrophilic, polar, where the phosphate heads are, but they are non-polar here in the middle where the fatty acid tails are. So for this protein to be embedded in the cell membrane, I need polar pieces here and here so they are happy to sit next. I shouldn't say happy, like molecules can't be happier said. It's just more stable. So they're more stable sitting next to the different polar pieces. If I have polar here and polar here, it's very stable sitting next to each other. And then on the middle, this is where we have our hydrophobic fatty acid tails. These amino acids need to be nonpolar hydrophobic in order for there not to be any repulsion. For our membrane proteins to stay stuck there, we need the matching polarities, non-polarities. Sometimes we can take our special amino acids and edit them a little bit to make them more polar. So this is proline. Proline is super important in our collagen. After a while, we can make hydroxyproline by adding this hydroxyl group. Hydroxyl groups are pretty polar, and this makes proline a little bit more polar. When our collagen starts to degrade, it turns into hydroxyproline instead of proline. And this is one of the things that diagnosticians will use to see if we have issues with our collagen. So here's another example of a membrane protein. This one is called a channel protein. Channel proteins literally are tubes that allow stuff to go through the membrane. We only have like a slice of it, but it basically wraps around. So literally like a tube, and things like sodium ions could flow through the tube in and out of the cell. So let's figure out which pieces and parts of this protein are hydrophobic and which pieces and parts are hydrophilic so that we know what kind of amino acids we would find in the different parts of the protein. Good. Remember that this whole thing is called a phospholipid. The head part, the phosphate heads, are hydrophilic. They're quite polar. The tails are fatty acids. They are non-polar. They are hydrophobic. So this chunk of the protein, these amino acids that make up this piece of the protein, since they're sitting next to all of the non-polar hydrophobic fatty acid tails, this part of the protein needs to also have non-polar hydrophobic amino acids. This piece, these pieces of the protein, the chunks that stick out of this cell, the chunks that are next to the hydrophilic phosphate heads, these all need to be polar amino acids, hydrophilic amino acids, to make up these pieces of the protein. Since we're sending sodium through this channel, the inside of the channel also needs to be hydrophilic. It needs to be polar, so the sodium is not repelled. So kind of cool how we can arrange all of these different amino acid properties into one protein so that we can be non-polar and polar all at the same time. We mentioned a couple slides ago that there were like 20 or 21 amino acids that all living things, almost all living things, use to build their proteins on Earth. And there are some reasons for maybe why there are only 20-ish that all of us use. It might be that those 20 were the ones that actually existed on the planet when life first evolved. Those were the only ones that were available to us. Maybe they're the best ones, or maybe we had some common ancestor that happened to use those amino acids, and so that's what we all use, since we've all descended from that one common ancestor. You might be wondering, how could we possibly have so much variation in our proteins if we only have 20 amino acids to pick from? It has a lot to do with the order of the amino acids. So let's do some fundamental counting principle math. I'm doing it sketchy style, so definitely there are some errors in this math, but stick with me anyhow. So we've got a dipeptide. Dipeptides are 2 different amino acids. If I have 20 possible options for my first amino acid, and I also have 20, because I could do like proline, 20 different amino acids for the second spot in my dipeptide, I'm going to multiply those two. There are 400-ish, like proline versus proline. There's some error there, right? Stick with me though. We're going to pretend like it's 400-ish different options for dipeptides. That's a lot of different dipeptides. What if I tried tripeptides? 1, 2, 3 amino acids. I have 20 options. I have 20 options. I have 20 options. Again, there's some repeats that we'd have to throw out. But basically, we have, oh my goodness, 8,000 different tripeptides. If I'm going with 10 amino acids, that's going to be 20 to the 10th power. That's a very large number. So we can put together some crazy wide variations with our proteins because of the different numbers different properties of our amino acids. The order of the amino acids in our proteins is coded for by genes, DNA, and our nuclei. We humans synthesize about 18,000 different proteins. This is known as our proteome. Proteome is the collection of proteins made by a cell or a system or an organism. The human proteome is just about 18,000 different proteins. All of our cells have 10,000 of those proteins in common. The other 8,000 are for specialized cells. So we definitely don't want our brain cells making the protein that makes hydrochloric acid like our stomach cells do. We want to keep that stomach acid in our stomach. We don't want that in our brains. The information for producing those proteins, of course, is coded for in the genes and the DNA sequences. Interestingly, only about 1% of our human DNA codes for protein. We call that the open reading frame. The rest of our DNA does other stuff. We didn't always know what that other stuff was, so for a long time, when I was in school, we called it junk DNA. We know now that it's not junk. It does have a lot of functions, but there's really only like 1% of our DNA that actually codes for protein. Only about 1% is known as the open reading frame. Again, proteome is the collection of all the proteins made by a cell or a system or an organism, and it is unique to each individual. Even amongst identical twins, they will have different proteomes. We can know what a proteome is because we can use this amazing piece of technology, gel electrophoresis, to isolate, separate, and then analyze all the proteins from a cell or a tissue or an organism. The proteomes are more unique than our genomes. So the genome is a collection of genes in an organism. The proteomes are more different than our genes are different. And some of the reasons, so we have different cells that'll make different proteins. So even though all the cells in our body have all of the same genes, we don't turn on all of the same genes all the time. And so different cells will make different proteins. And then even the same cells will make different proteins at different pieces of their life cycles, because as we age, we need different proteins. It's pretty amazing how we can turn on and off our genes to have the proteins that we need. When we build our amino acid chains, first we're going to call those polypeptides. Once we have some functional working polypeptide units, we're going to rename those proteins now. Some of our proteins are composed of only one polypeptide chain. Lysozyme here is a tiny baby little protein. Its job is to chop stuff up. Some of our proteins are composed of many different polypeptide chains that are stuck together. Hemoglobin is an example. Hemoglobin is a protein that carries oxygen around on our red blood cells. It's actually composed of 4 different polypeptide chains. There are 2 blue guys, 2 red guys. We call them alpha and beta, but there are 4 subunits that make up hemoglobin. Proteins have different 3-dimensional shapes. We're going to call that their conformation. The conformation of a protein gives it its function. The shape is determined by the amino acid sequence. And so when we look at the different shapes of these proteins, these are the same ones that we looked at very early on in this slideshow. So these different protein shapes are determined by the amino acid sequence. So Rubisco and collagen have very different amino acid sequences, which is why they have very different shapes. That amino acid sequence that determines the shape of a protein is known as the primary structure of a protein. The primary structure is its amino acid sequence. The amino acid sequence of a protein is its primary structure. We can see here that we have an amino acid. Here's the amino end. Here's a carboxyl group, and here's the next amino group. So here's our peptide bond. Here's the start of the second amino acid. Here's another peptide bond, another amino acid, another peptide bond, another amino acid. Whatever that sequence of amino acids is, maybe this one is methionine, and this one is proline, and this one is leucine. Whatever that amino acid sequence is, that is going to be the primary structure of the protein. That primary structure is going to determine all the other levels of structure. There are 4 total levels of structure of proteins. The secondary structure comes in 2 forms. It can be either alpha helix or beta pleated sheet. Alpha helix or beta pleated sheet. Both of these are held together by hydrogen bonds. Hydrogen bonds are forces of attraction due to a partial positive thing someplace and a partial negative thing someplace else. How that works in our amino acids, our amino group has that lonely hydrogen. It's going to be slightly positively charged. The carboxyl group of a different amino acid is going to have that double bonded oxygen. It's partially negatively charged. And we know that opposites attract. And so we're going to end up with a lovely force of attraction between the hydrogen of one amino group and the oxygen of the carboxyl group of a different amino acid, and they stick together like little magnets. Depending on the order or the primary structure of the polypeptide, we're going to get different forces of attraction in different places. And then we end up with either this curly Q thing, alpha helix, or we end up with these big folds, beta pleated sheet. But then there's more. Tertiary structure, this is more three-dimensional structure. So tertiary or three-dimensional structure, this is going to be like big folding, like whole entire chunks of this polypeptide that might have some curly Qs or some folds in it. The whole thing is going to fold in on itself. And then we're going to hold it together with some more hydrogen bonding. But we're also going to add in some ionic bonds and some covalent bonds. So primary structure is held together by those peptide bonds. Secondary structure has only hydrogen bonds. Our tertiary structure has hydrogen bonds and ionic bonds and covalent bonds. It's a special kind of covalent bond called a disulfide bridge. That disulfide bridge is formed between two cysteine amino acids. Cysteine has sulfur in it. So if I have two cysteines, I have two sulfurs, hence the disulfide. So that's the tertiary or more three-dimensional structure of a protein. But wait, there's one more. We also have quaternary structure. If we have multiple polypeptides that get stuck together to make the functional protein, we have quaternary structure. Quaternary, or that fourth level of structure, we're going to have multiple polypeptides, each of them with its own primary, secondary, and tertiary structure, then get stuck together. This is hemoglobin. Hemoglobin has let me pick a different color. 1, 2, 3, 4 different polypeptides that then get stuck together with some hydrogen bonding and some ionic bonding and some covalent bonding, just like the tertiary structure. Cool beans, right? That tertiary structure, that three-dimensional folding, is not the same in all proteins. And depending on the level of that three-dimensional folding, we're going to end up with proteins of different shapes. Globular and fibrous are the two big categories of protein shapes. Globular proteins are more rounded. They are not spherical. They're not like perfect little spheres, but they're more rounded. And they tend to be more water soluble. They dissolve better in water. They have more three-dimensional folding, more tertiary structure. They have more hydrophilic amino acids on the outside that interact with the environment, and then the inside, the ones that are kind of embedded on the inside of the protein, those are more hydrophobic so that they can stick together. Some examples of globular protein, rubisco and insulin and rhodopsin, these guys are all a little bit more rounded. Our fibrous proteins, on the other hand, are going to be a little bit more stringy. So fibrous proteins are insoluble in water. They have less of that tertiary three-dimensional folding. They're long and stringy, like collagen and spider silk. They're just a little bit more stringy. Our globular proteins tend to be more metabolic in function. So these guys are enzymes. Our fibrous proteins tend to be more structural in function, so like the collagen that holds our skin together. The ability of a protein to do its job is very dependent upon its shape. So structure and function, very interdependent. If we mess up a protein's shape, we're going to mess up its ability to do its job. Messing up a protein's shape is known as denaturation. The disruption of folding patterns, basically we unshape the protein's shape. We can do this with heat. If you have ever cooked an egg, you know that the protein in the egg definitely changes with that heat. We can also change the shape of proteins with pH, with salinity. That's the saltiness of a solution. So these things can mess up the shape of a protein and then mess up its job. Sometimes we can renature the protein, we can put it back into its shape, but not always. Sometimes, like that denaturation is like it's done. That protein is dead forever and ever. But if we have a denatured protein, it cannot do its job. For the protein to be able to do its job, it has to have the correct shape. What are some of those jobs of proteins? Well, here are lots of examples of protein functions. One is structure, so collagen, spider silk, histones. These are the proteins that our DNA wraps around. These are all structural proteins. We can have proteins function as hormones, insulin and glucagon. We'll talk a lot about these when we get to our unit on physiology. Immunity is carried out in part by proteins. Immunoglobulins, which we also call antibodies, are super important for our immune system. When we clot blood, when we get a cut and we have to form a scab, that's done by proteins. Transportation of some things like hemoglobin carries oxygen around the body. This is an important transport protein. We have sensory receptors, rhodopsin in our eyeballs, helps us to see. Movement, our muscle is so much protein, actin and myosin, we talked about that, titin. And then we have enzymes, our metabolic proteins. Rubisco is one, catalase, amylase. Generally, if it ends in an ase, it is an enzyme and it carries out metabolism for us. We humans love to put proteins to work for us in technology. We put proteins into detergents. I don't know why this entertains me so much, but I find it golden. Bioenzyme power. So we've got some proteins in our soap. We also put proteins to work in diagnostic tests sometimes. This is a pregnancy test. There are antibody proteins on that stick. Helps with the color change so that we can diagnose pregnancy. Pharmaceuticals, of course, we use proteins for medicine. Here's some insulin. People who have type 1 diabetes need to inject this protein into their bodies to help stay alive. And then our food, we can actually use proteins like lactase to break down the lactose in milk for people who are lactose intolerant. And we can make lactose-free milk by using our enzymes. And my friends, we have come to the end of this video lecture on proteins. We accomplished our objectives. We talked about proteins, our chains of amino acids held together by peptide bonds. We know how to draw a dipeptide and show that peptide bond. We talked about how amino acids have different properties. Some are polar, some are non-polar, and those properties are conveyed to the proteins. So we have proteins that have different functions. Proteins have those 4 levels of structure. Primary structure is the amino acid sequence. Secondary structure, alpha helix or beta pleated sheet held together by hydrogen bonds only. Tertiary structure, that 3-dimensional folding held together by hydrogen bonds, ionic bonds, and covalent bonds, also known as disulfide bridges. Quaternary structure in some proteins that have multiple polypeptide units. Quaternary structure also held together by hydrogen bonds, ionic bonds, and covalent bonds. Proteins have different shapes. They can be globular or fibrous. We can actually unshape the shape of a protein. That's called denaturation. And we know that the function of a protein is related to its structure. Good work today.